Bismillahirrahmanirrahim. Assalamu alaikum. Now, from today onward, I'm going to start a series of videos or vlogs or lectures. My objective is to let our students, MPhil, MSc, even BSc and PhD, to learn SAS software, which is a statistical software, and that is what I recognize is one of the best software I have worked with right from my PhD time when I was doing PhD. So this is one of the best software. I used various softwares. I used MStatC, I used Minitab, I used SPSS, and I used SAS. So what I found the best in SAS. Why I chose SAS to continue working and analyzing data for my research, for my student research, and whosoever used to come to me for help, to seek help for their master degree or PhD degrees. The student used to come to me for their data analysis. So I preferred to use SAS rather than SPSS. So I recognize that SAS is a very, very flexible, handy software. SAS, it's very flexible. I did not require a lot of manipulation with the data arrangement. This SAS is very flexible, so I preferred SAS. So this is what I will recommend you people to use SAS. I used R as well. I got a training on R as well. So the first thing I would like to let you people know is that the data in Excel sheet is very, very important. So today I'm going to let you people know that what are the requirements of SAS as far as data entry of your research data into Excel sheet is required. So that is what I'm going to talk today. I hope you people will understand it very easily, and you can make your notes if you want, but anyway, these videos will be available on the YouTube, and hope you'll get a good benefit out of it. So let's start with the data. I'm going to present here one of my students' data. So I will let you people know that how the data is entered into Excel sheet, research data. So this time I'm going to tell you about the experimental research data. That we do experimentation, we get the data, and how to enter that experimental data into Excel sheet. That is what I'm going to tell today. In next few videos, I'll be talking about epidemiological data, that is observational data or survey data, and even I'll talk about the molecular biology data, especially the binary data, which we get from different molecular biological research. So three kinds of data I'll be letting you people know, and I will let you know what are the best software to handle what kind of data. So SAS is very good, but for certain kinds of work, I use another software. So I'll let you know that also. That is quite handy as well. So let's start with the how to enter the data in Excel sheet, the experimental data of agriculture fields or veterinary field or human sciences or zoological sciences. So let's start with. Now here you are seeing an Excel sheet. This is the data of my student. Remember, this is how data has to be entered. The top row should indicate the names of the variables. Different variable names, they are given on the top row. Now I have fixed this top row, if you can see. I'm moving the data down so that I should see what variable is having what value. So I can see all the data here, okay? So here the top row is fixed. In the top row, the names of the variables are given. The names of the variables, they are comprising of 8 alphabet. You can use capital and small letter, but never give space between the two words. Like if look at here, it is liver index. Now, this is sheet number 1 where our main data is entered. And here is the sheet number 2 where this is for making notes what abbreviations are used in sheet 1, they are noted in sheet 2. So these are basically notes. Group 1 is what, group 2 is what, group 3 is what treatment has been given or to the group 1, 2, 3, 4, likewise. So you can see here, now it is better to record your notes on the sheet 2. Never use loose paper, register, or any other such documents because it can be lost. Now, here if the data is lost, everything is lost. But if a sheet is there, the data is there, its notes are also there. So you can have the notes along with your data. Now, the abbreviation used, they are written here, like live weight, LWT, likewise, okay? Now, so, like here you are seeing liver index, so it is index of the liver. Liver weight divided by live weight into 100, that will give our liver index. Anyway, so this way you can record, you can calculate your liver index and all different parameters. As I told that the name of the variables are given in the top row with maximum of 8 alphabet. You can use the large and small letters, and beneath in the column, in each column the data is entered. Now here it is sample 1. Sample 1 can be a date. So up to here it is sample 1. There are 5 groups. So for each group, 5 birds. Basically it's a data of 5. Now this trial was conducted on broilers. So there is a data of broilers, 5 birds in each group. Here is the 6th group for sample 1. It's the sex of the bird is also noted. Remember this, it's very important. The live weight and the carcass weight, 1 is indicated for female. Here you can see 1 is for female and 2 is for male. And you can see here the live weight of female is lower than that of male. So same is for various enzymes and other parameters. There is always a variation on the base of sex. So if you have not recorded the sex in the experimental data, whether it is human data or whether it is animal data, the statistical results which you are going to get, they are not the true. That will not reflect the true picture. And this is why the different variation in different research conducted in Pakistan, in China, in USA is there. Because in advanced world, in the developed world, everything is recorded and it's the confounding effect is nullified and then data analysis is done. While in developing world, these things are missing, lacking, and this is why the accuracy in the results is not there. That is very, very important. So you can see how the data is recorded. This is group 1. Because in each group there are 5 birds, so the 1 is indicated for 5 times. And this is group 2, again 5 times, because there are 5 birds in this group as well. So, and here are the values for each bird. So this is how the data is recorded. This is for sampling 1. Here is the sample 1. It can be a date. Here is the sample 2, which you might have got after 2 weeks or 1 week's interval. Second sampling has been done. Third sampling has been done. Like here, third sampling has been done. There may be a missing value. Leave it as such, no problem. So this is very important really to understand that how data is entered into the Excel sheet. Now you can see here how data is put into Excel sheet. This is very, very important, and a proper data recorded sheet is required for SAS. Now I use SAS, so we will look into how the data analysis is done in our next video. Here in this video, my main objective was to let you people understand how data is put into Excel sheet. I hope you have understood it very well. I hope this video has done good for you, and lots of good. Anyway, this is how the Excel sheet should look like. So I have shown you how to enter the data in Excel sheet. This is very important. I do hope you people have understood it. This is experimental data, and if you find any question, if you have any questions, you can contact me through WhatsApp. I will be available to all of you. Whosoever want to ask any question about it, I'll be there to help you people. I do hope you have understood it very well. In next vlog, I'm going to let you people know how to use SAS and how to analyze data in SAS. Let's hope good for everything, and I do hope you have a good learning today. Thank you very much. See you inshallah next time. Allah Hafiz.